Now is the winter of our discontent made glorious summer by this widely awaited closing lecture by Manuel Serrano. I will not introduce Professor Serrano because we all know him very well. So, after all, we came here, all of us, to listen to him, not to listen to me. I will only mention one thing that Manolo and I have in common, and this is the unusual Wikipedia page. If you type Felix Goni, you will find my image, together with the biography of a Catalan terrorist who died in the 80s. But then type Manuel Serrano, and even more Manuel Serrano Barcelona, and you will find that he is a famous hairdresser. Anyway, so it's better if I don't go into these details, and I will just leave the floor to Professor Serrano. I always say, he is the best science speaker in the Spanish language that I have ever met. Unfortunately, today we cannot profit from this. It will be in English, but remember. Okay, Manuel, please.
I also have a personal feeling of closing because, as some of you may know, I will move to Cambridge, UK very soon in January. So I have this feeling that maybe this is my last participation in a conference in Spain for a while. But I will keep in touch, of course. And I feel a bit emotional today. Thank you. I'm going to talk about, this is going to be the topic of my talk, about cellular rejuvenation by partial and reversible reprogramming. I will explain in a second what I mean by this. Before that, I want to put this research in a wider context, because sometimes I have the feeling that the scientists that are working on aging, we don't communicate very well why are we studying aging, and I will tell you why, in my case, we are studying aging. First of all, because aging is the main risk factor for the large majority of diseases. The main risk factor for diseases is not our genetic makeup, is not our lifestyle, is not our exposure to toxics or agents. The main, by large, is aging. However, is one of the poorly understood risk factors for diseases. I think this is a paradox. We have to understand what is aging that makes us vulnerable to so many diseases. In addition, particular diseases today are considered by many, including by myself, as a process of accelerated focal aging. This is true particularly for chronic and degenerative diseases. These are the ones that are typical of old age. Lung fibrosis, kidney fibrosis, steatosis, atherosclerosis, neurodegeneration, all those diseases can be considered somehow as an accelerated process of aging in a particular tissue. So again, understanding aging, we understand the predisposition, and we may also understand the diseases. And in my case, we are studying aging because this may open new therapeutic opportunities to treat or prevent diseases. I'm not thinking about treating the entire population. I'm not thinking about treating healthy old people to be younger or to live 200 years and all these things that maybe is a motivation for others. But not in my case. We have a more humble or realistic goal, that is to treat or to improve the life of people with debilitating diseases. A number of years ago, actually very soon it's going to be 10 years, and we are preparing now with Carlos and the rest of the authors, we are preparing the 10-year update of the hallmarks of aging with new hallmarks like inflammation, like the microbiota. But I show you this because I'm going to talk about one aspect or one strategy to rejuvenate cells. I don't mean to be the best, it's just one strategy. It is a strategy that goes directly to the nuclei, to the DNA. And some people think that the DNA is like the brain of the cells, and by rejuvenating the DNA, somehow this communicates to a rejuvenation of the entire cell. So I'm going to talk about reprogramming as a tool to rejuvenate cells. What means cellular rejuvenation is always a bit tricky. We know, we feel when we become old, and we can know when a person is younger or older, but to distinguish a cell that is older or younger is a bit more abstract and difficult, but I will go into that. Okay, so reprogramming. Reprogramming was discovered in 2006 by Yamanaka, and he demonstrated that introducing 4 transcription factors, OSKM, I will abbreviate them as OSKM, when they are introduced ectopically into any kind of cells, from hepatocyte, lung cell, any cell, these transcription factors are able, first of all, to shut off the identity of the cells. They become undifferentiated, and eventually they acquire a new identity. So we start with one identity and we arrive to a different identity that is embryonic pluripotent stem cells. One thing that is interesting that has been discovered in the last years is that these transcription factors that normally are positive transcription factors, they activate transcription. What they do in the cells is not to activate transcription. They are actually repressing transcription. So the first days, this takes about 10 days. The first 5 or 6 days, what is happening is that the cells are repressing transcription, particularly of cell identity genes. So the cells become de-differentiated. They become something poorly defined. And then eventually, now, these transcription factors activate transcription. So there is a wave of repression and then a wave of activation of new cell identity genes. We start with one identity, cells lose their identity, and they acquire a new identity. This is full reprogramming. A number of years ago, or almost 10 years ago, Maria Abad in the laboratory generated mice that have become very useful in the field, that we call them reprogrammable mice. In these mice, she introduced a transgene where the OSKM cassette is induced in all the cells of the body, driven by a ubiquitous promoter. And in this way, she was able to observe events, the same thing that happens in cells in vitro, also in vivo. I have to say that this process becomes more and more inefficient as it progresses. So the number of cells that they differentiate maybe is 50%. The number of cells that arrive here is maybe 1%. There are many cells that are lost in the process, in this transition. The same happens in the mice. Only a fraction of cells they differentiate, but this is something remarkable. Now this is a mouse where you can induce dedifferentiation in the kidney or dedifferentiation in the liver, in many tissues, and it's a tool to understand these cellular transitions and cellular plasticity. We like to call it cellular plasticity. These factors eventually are able, in a minority of the cells, to generate IPS in vivo. We can detect them in the blood, and we can detect them in situ in tissues. And these IPS will do in pluripotent stem cells. They generate teratomas. They generate a type of disorganized embryo. And this was very good news, because only by showing this, people scientists in the field were convinced that we were actually doing reprogramming. Otherwise, they were thinking, well, who knows what is happening in vivo? Well, we have teratomas, therefore, full reprogramming has happened. Now the field, as I will show you, has shifted the focus from the full reprogramming, from the IPS, or from the IPS and the teratomas, to these intermediate states, because it is believed that here is where things, interesting things are happening. To start with, because we don't want to change in vivo the identity of the cells. If we have a liver, we want to improve the function of the liver, but we don't want to generate pancreas in the liver, for example. We want cells to maintain their identity. And for that reason, the focus is here, before the process is irreversible. I'm going to show you here an example of histology of the pancreas. It's one of our favorite tissues to study reprogramming. This is a little bit of normal-looking pancreas with the acinar cells here. And this is in the reprogrammable mice, when the process is in the intermediate phases, how the pancreas looks. It looks completely abnormal and very heterogeneous. There are cells that look, they have different morphologies. They don't look anything similar to the normal acinar cells. And actually, as I will show you in a second, they have lost the identity of acinar cells. And they are becoming something that is poorly defined, and still we don't understand very well what it is. Very heterogeneous. Something similar happens in other tissues, in the intestine, in the kidney. And that's it for now. So, the reversibility. This is what I have explained. But now, what many scientists are doing is to interrupt the process of reprogramming here. Instead of expressing OSKM all the way, OSKM is expressed half the way. Instead of having the doxycycline to induce the transgene for 10 days, it is exposed to the mice for only 5 or 7 days. And then it's removed. And what happens is that cells lose their identity, but they still retain epigenetic memory, or they have not lost identity completely. And when the doxycycline is removed, the cells reacquire the same identity. So you may think, well, where is the reprogramming? No, there is no reprogramming. The cells start with one cellular identity, and they go back to the very same cellular identity. It's a transient process of dedifferentiation. They dedifferentiate and then redifferentiate. And the surprising thing, as I will mention, is that in this process of losing their identity and reacquiring the same identity, now these cells here, in this new start, they are younger according to cellular markers of aging. If these cells are old and they have markers of aging, different types of markers, particularly in the DNA, epigenetic marks, when they go through this process, they look younger. You may consider this like a new start. They lose their identity, but not completely. They re-establish the same identity, and now they are younger. It's a fresh start for these cells. And this is what we call partial because it's not complete, partial, and reversible because it goes back to the original identity. Partial and reversible reprogramming. Sometimes I will say, just for brevity, I will say reprogramming. But when I say reprogramming from now on, I refer to this. The laboratories of, particularly Juan Carlos Izpisua Belmonte was the pioneer. He published the first paper where he showed this rejuvenation potential of partial reprogramming. But there are other papers reporting similar findings, making more functional tissues in old mice. For example, in this paper, in Science, they rejuvenated the heart. in all mice, so that now the rejuvenated hearts, they are able to sustain an infarct better than an old mouse. This is with macular degeneration, and Juan Carlos has reported for diabetes and for a number of diseases. When this process of partial rejuvenation is done in cycles, multiple times, the mice are younger according to this criteria. Our little contribution here has been to use a single cycle. All these papers are using multiple cycles. Why? Because, as I mentioned in the beginning, this reprogramming is inefficient. Only a fraction of cells benefit from this. There are many cells that don't progress in the process of reprogramming. By doing multiple cycles, in a way, you are increasing the percentage of cells that rejuvenate. This is the idea. This is the idea, and it has not been formally demonstrated. So we wanted to do just one cycle of OSKM, and of course, one cycle is not sufficient to make the mice look younger. But if we go deep into the cells, we can detect evidence of rejuvenation. I'm going to show you, we have studied a number of aspects. I'm going to show you just one example with the transcriptome. We did the RNA-seq of the young pancreas and old pancreas, and here the entire transcriptome, and then we identify which genes change with aging. The fraction of genes that change their levels with aging is small, is 5%, 1%, is not very high. If we take those genes, of course, by principal component analysis, the old mice and the young mice are clearly separated. This was the condition. And now we can ask what happens with the old mice when they are exposed to one period of partial reprogramming. And they are in between. They are not fully rejuvenated according to the transcriptome, but they are partially rejuvenated. Of course, there is heterogeneity, and this has not rejuvenated as much. And another interesting point is that when we ask which out of these few hundreds of genes that are rejuvenated, to which categories they belong. The number one category is the mTOR signaling pathway. This is interesting in the field of aging because this is the signaling pathway that universally across species increases with aging. This is a transcriptional signature that in all animals, in yeast, in Drosophila, in C. elegans, in mammals, goes up and up and up and up with aging. So when we do this, this signature goes back to the younger configuration. But let me move now into work that we are doing now that is unpublished. So now I'm going to talk about two stories with unpublished work. The first one is trying to understand these intermediate states, because this is very heterogeneous and is not well defined. And this is what most people are interested in, because this is where the magic of erasing the old age and establishing a young configuration is happening. So to understand this, well, first of all, another thing that is interesting is to know how far this process of reprogramming has to go before we enter into the risky area of pluripotency, because pluripotency will give rise to teratomas. We don't want to get teratomas, we just want to get the rejuvenation. So we have done single-cell RNA sequencing of the tissues of the pancreas. I'm not going to show you all these UMAPs and all that, I know it's boring. I'm going to show you the final data, the conclusion, and some data is work that we have done in collaboration with Paco Real at the CNIO. And we call this the roadmap of reprogramming in vivo. First of all, identity, cellular identity. I said that the first thing that the Yamanaka factors do is to induce de-differentiation, loss of cellular identity. So we use, this is a single molecule fish. And we tested this marker of cellular identity, carboxypeptidase A1. It's an enzyme, a gene typical of us in our cells. And when we look at these pancreas that are in the middle of reprogramming, what we find is that there are areas that have lost this gene. We don't understand why there is these regional patterns. Instead of having cells intermingle, some of them are de-differentiated and others retain differentiation. We find patches. We don't understand why. Probably there is some cell-to-cell communication. But these are the cells that are undergoing reprogramming, and these are the cells that somehow have resisted, became refractory to the reprogramming. So retention of cell identity, and these are the ones that are reprogramming. We did RNA-seq of this situation, and we found markers of these cells, and we established a trajectory. And this will be the summary. When the Yamanaka factors are activated in a tissue, all the tissue enters into a state of stress, and we can detect that with markers, and we call this pre-reprogramming. The cassette is induced in the large majority of the cells. And then these regional patterns are established in vivo in the tissues. There are areas with high AP1 activity, stress probably, that they do not reprogram. They go back to the normal identity and they resist reprogramming. And there are, these are the cells that are here. And then there are other cells that enter into this transformation. They lose their identity, they lose these markers of cellular identity, and they start to activate unusual genes. For example, this mucin is typical of the intestine and of the lung. Now it's expressed in the pancreas. And many other markers. I'm putting here the examples that we have studied more carefully in situ. When the process goes and advances even more, other proteins that are very unusual in the pancreas, like keratin 14 is never expressed in pancreas, starts to be expressed. We think that this is recapitulating to some extent the development. Here the cells look like endodermal precursors. And here the cells start to look like ectodermal precursors. If you are familiar with development, the first identity that the cells have in the embryo is ectodermal, and from the ectoderm, the gastrulation generates the endoderm. So maybe these cells are recapitulating, are going back in developmental time. And then eventually they reach pluripotency. One marker that is very interesting and is here is LY6A. I will dedicate a second to this marker. This is very famous because it's a marker of many somatic progenitor cells, particularly in the hematopoietic system. And here is appearing in these cells that are reprogramming. These markers are also conserved in different tissues undergoing reprogramming. When we reprogram the stomach, we see, this is RT-PCR, we see more or less the same markers that we found. Here is a list of 20 markers that we identified in the pancreas. They are also found in the stomach, and the same have done in the colon. And also in vitro, in cells in vitro, the same markers are upregulated. Each lane is one mouse. Injury is not activating the same program in the pancreas, although I will talk about the injury in a second, about the relation between reprogramming and injury. With LY6A, we have the advantage that there are many tools, and we can detect this intermediate reprogramming by immunohistochemistry. And here we can see in the pancreas, the regions that are being reprogrammed express this marker, also in the stomach or in the colon or in the intestine. So we think that now we have a method to capture these intermediate cells and we can study this rejuvenation in these cells. And 5 more minutes to talk about another topic that we are working on, that is these extrinsic boosters of reprogramming. Why are we studying this? I particularly don't think that introducing Yamanaka factors into people is going to be a viable strategy. Very few people think that. Of course, it will be extremely dangerous, and it's uncontrollable. There are many, many concerns about that. Although some scientists, they are following that path, but not the majority. What we are trying to understand is how the cells arrive to this state of intermediate or dedifferentiation. We want to understand which factors, external pharmacological interventions, can improve this reprogramming driven by the Yamanaka factors. Everything that we have done is driven by the Yamanaka factors, and we are potentiating that in the hope that eventually we may someday get rid of the Yamanaka factors and do the reprogramming without any genetic manipulation. That would be the ideal, to do everything pharmacologically or nutritionally. In the past, we have published how recombinant IL-6, which is an approved medicine, or palbociclib is used for cancer in all the hospitals for breast cancer. How these drugs boost the reprogramming in vivo. So here we have one way to improve reprogramming. Also, if we deplete temporarily the natural killer cells, we also increase reprogramming, because the natural killer cells detect these abnormal cells, these intermediate dedifferentiated cells. They detect them as something weird and they kill them. So if we deplete NK cells, we have a lot of reprogramming. And vitamin B12. I'm going to talk about this briefly because I know I'm in an audience of experts in metabolism, in biochemistry, and I am not. So I want to present this perhaps to get good ideas or good hints. Marta did an unbiased, I want to emphasize this, it was an unbiased screening for nutritional and metabolic dependencies of reprogramming. She induced reprogramming in the mice and she studied the metabolism. And doing that, the number one metabolite that she found was vitamin B12. He analyzed the levels of vitamin B12 in the serum of the mice. These are mice that are fed with normal food, and the normal food of the mice contains a lot of vitamin B12. It's a vitamin, is required, is present always in saturating conditions. So it was very strange that these mice that are undergoing reprogramming, they have a reduction in the levels of vitamin B12. Very unusual. This gave us the vitamin B12 is necessary, I'm simplifying for the experts, apology, but is required almost for just one metabolic reaction, that is methionine synthase. The production of methionine, and methionine produces S-adenosyl methionine, which is the precursor for all the methylation reactions. This, for sure, is all. All the methylation of the DNA, of proteins, everything of lipids is done with S-adenosyl methionine, and that requires vitamin B12. We measure a signature of methionine starvation, and she found that the tissues that are undergoing reprogramming, this is in the pancreas, the tissues that are undergoing reprogramming, they have activated this signature of methionine starvation. The pancreas of these mice is starved of methionine, whereas the kidney of the same mice, and the kidneys in this case are not being reprogrammed, they don't have this starvation. So in the same mice there is a selective demand of methionine in the pancreas, and the pancreas feels that needs more and more and more methionine. The transporters for vitamin B12 are induced, many other things are induced that I'm not going to show you. This is the methionine cycle. We look if this is true also in vitro, because in vivo, of course, things are super complicated. And in vitro, it happens the same thing. Here we are counting iPS colonies. These are fibroblasts in vitro with culture medium, which always contains a lot of vitamin B12. And Marta found that if she adds even more vitamin B12, there is more reprogramming. If this enzyme, the one that produces S-adenosylmethionine, is inhibited, then it is reduced. And if she complements directly with S-adenosylmethionine, there are a lot of colonies. So in a cell-intrinsic manner, in vitro, the cells that are reprogrammed, they require a lot of methionine and vitamin B12. Also in vivo, these are mice supplemented with vitamin B12. They have more reprogramming. This is measured by dysplasia and by this marker NANOG, more reprogramming. This was unexpected. These mice are eating a lot of vitamin B12 in the food, but if you put more, there is even more reprogramming. Of course, we thought about all the remodeling of the DNA that is happening in these cells that is going to require a lot of methylation. Methylation of histones and DNA is dynamic. It's not just to put methylation and it stays there forever. It has to be maintained all the time. And we asked which methylation mark is sensitive to the levels of vitamin B12. We did an array of 16, all these epigenetic marks, and we asked which epigenetic mark is changing when we put extra vitamin B12. And there is one that consistently appears, that is here in red, is the one that increases the most when we put extra vitamin B12, and is keratin 36 trimethylation. I didn't know at that time much about H3K36. Is not one of the most famous marks because it is not in the promoters or in enhancers. It is in the gene body, and therefore has the attention of most epigenetic experts has been in the epigenetic marks in promoters and enhancers. K36 is a mark that is introduced in gene bodies as the RNA polymerase is traveling in the active genes. So we check K36 in the gene bodies. This represents the gene bodies. This will be the promoter, and these are the gene bodies. And these are the levels of K36 in normal MEVs with vitamin B12. Adding vitamin B12 in a normal condition, no change. And these are the MEVs that are being reprogrammed. I hope you can see that there is a global depletion in K36. So the global levels of K36 trimethylation are reduced during reprogramming. And if we add vitamin B12, it's rescue. The absence of K36 has an impact on what is called cryptic transcription. In gene bodies, sometimes there are places for RNA polymerase in the middle of the genes. And these cryptic sites have to be silenced because of keratin 36 is trimethylated. And it is known, there are many papers, it is known that the absence of K36 or the reduction in K36 increases the amount of cryptic transcription. These are transcripts that should not occur, and now they are happening. So we have measured that as well, and we have found that globally in the, so I lost well, these are the MEVs with the reprogramming. They increase the amount of these cryptic transcripts. These cryptic transcripts, of course, are going to produce aberrant proteins and to interfere with many processes. And with vitamin B12, what we are doing is to improve the accuracy of transcription during reprogramming. And with this, I'm going to wrap up. Because one of the things that we want to do, and this is still far from publication, is we want to make a parallel between reprogramming and tissue repair. The field of reprogramming has sometimes been criticized as being something very artificial. That in a normal process, when in physiology is this differentiation and rejuvenation happening? This is a difficult question to answer, but perhaps it's happening during tissue repair. And the number of parallels between tissue repair and this partial reprogramming are increasing in the literature. Let me show you here one example. We're studying repair of ulcers in the colon. These are ulcers that we induce with DSS, dextrane. This is a very well-established model of ulcers in the colon. There is a paper, very nice paper, reporting that when the colon is repairing, it activates fetal signature. Actually, the title of the paper is a fetal signature. Remember what I said about reprogramming that induces something that looks like endodermal precursors and then ectodermal embryonic precursors? Well, they find these endodermal precursors in the repairing colon. No Yamanaka factors here at all. And the marker that they identify as the best one is LY6A. LY6A is also called SCA1. Sorry for using two nomenclatures. And Marta has found that during the repair of these ulcers, the amount of vitamin B12 is also reduced, the same as it happens during reprogramming. Actually, when this is the amount of SCA1 in the process of repair with DSS, this is amount of SCA1, this is the amount in the addition of vitamin B12, there is a little bit more, and this is during reprogramming even more. And if we supplement with vitamin B12, the repair process is accelerated. This is a score of the damage produced in the colon, and this score is reduced when we add vitamin B12 or when we add OSKM. If we combine them, there is no benefit. There is no further benefit. So, as you can see, there is a parallel between, in some aspects of what is happening during reprogramming and tissue repair. And I'm going to show you the last slide, which is a speculation. It's purely a speculation. There are many studies now in the field of tissue repair, in the skin, in the colon, in muscles, many, that are converging on the idea that the maintenance of a tissue in homeostasis is different from the repair of a tissue upon damage. When a tissue is damaged, the damage generates the differentiation and de novo emergence of progenitor cells. Some cells that were differentiated, they differentiate and are able to repair the tissue. It's like a facultative ability to generate progenitor or stem cells. And they repair the tissue. So there is a first step of the differentiation in tissue injury, and then there is a second step of redifferentiation. Sounds familiar? I had another slide here. Something didn't work, but it's not important. Remember what I said about reprogramming. In reprogramming, there is a first step of dedifferentiation and a second step of redifferentiation, and that is a new start for the cells and a rejuvenation. And there is this idea that perhaps when tissues are repaired, they are rejuvenated. It's paradoxical that you need a damage to rejuvenate. There is a work in bioRxiv from Vadim Gladyshev in the MIT measuring epigenetic aging in mice, and they have produced traumatic damage, traumatic bone traumas in the mice, and they show how these traumas, they induce an acceleration of the epigenetic age, and when the bones are healing, it produces a rejuvenation. Actually, the mice rejuvenate a little bit younger after the repair of the bone. So there is this idea, and I want to finish with this, that perhaps with the reprogramming, we are somehow having a glimpse of how tissues repair, and perhaps to understand this process of cellular rejuvenation. And with this, I want to finish mentioning some of the main characters in this story in the lab. Marta Kovacheva is here. And thank you for your attention. It has been an honor for me to have this opportunity. Thank you.